Good afternoon. I'd like to thank the organizers for the invitation to present my work here. My name is Ricardo. I'm a staff scientist in the group of Ben Engel in Basel. And today I'll be talking about HDCR, which is a filamentous enzyme from bacteria that can do carbon fixation very efficiently, and how we use it cryo-ET to study it. And towards the end of my talk, I will also show a little bit of some new membrane segmentation tools that we are developing in the group. So the outline of my talk is basically this. I will introduce HDCR and show you some structural insights into its function that we got using cryo-EM and cryo-ET. And then I'll present you the native structure of HDCR using in situ electron tomography. And the last part of the talk, I will talk about the new version of membrane, which is a deep learning tool that can do very accurately membrane segmentation. Okay, so I hope you all know that our planet is warming very rapidly, too fast, too much. And this is caused by human-made carbon emissions in the past 150 years or so. And in this context, our group studies the molecular basis of carbon fixation. How is it done in nature and in different ways? And we do this because we want to understand how our world is changing, and hopefully we can find some new tools that we could use against global warming. And today we are going to be looking at this organism, T. kivui, which was originally isolated from the bottom of Lake Kivu in Central Africa. And this is an anaerobic protein. It's an acetogenic, and it's thermophilic, so it has very stable proteins, which makes it an ideal organism for studying these things. And it's also a gram-positive bacteria with a very cool S layer. And HDCR is not the hardcore complex; it's the hydrogen-dependent CO2 reductase. This enzyme was discovered by the group of Volker-Müller at the University of Frankfurt, and it is part of the initial steps of the Wood-Ljungdahl pathway for CO2 fixation in these acetogenic bacteria. And it consists of 3 modules, basically. At the center, we have the electron transfer module, which consists of polyferric doxins. And this electron transfer module is attached to a hydrogen splitting module. So it basically steals electrons from molecular hydrogen. And there's also a CO2 reduction module, which is the format dehydrogenase, shown in green here, which is, so to say, the carbon fixation module. And this reaction works reversibly. And this enzyme is extremely efficient in converting CO2 and hydrogen into formate. And we would like to understand how are these reactions physically coupled. And why? Because if we understand this could have some very interesting biotech applications, for example, for CO2 capture and hydrogen storage. So our collaborators in Marburg, the group of Jan Schuller and his student Anuj Kumar, they set out to solve the single particle structure of HDCR. And basically, they solved the structure of very short filaments that they found on their grids. And this structure is resolved to about 3.4 angstroms. And this is how it looks like. So it turns out that HDCR is a filament. And this filament is composed, it's a heterohexamer. So there are 2 hydrogenases, the blue domains shown here, and one format dehydrogenase, the carbon fixing module, which is shown in green. And there are 3 different ferredoxins that form the backbone of this filament. And as you can see, there are many iron sulfur clusters which are shuttling electrons around the protein. And this was some parts of the map were quite difficult to model, and for that we use AlphaFold to help us. And the key insight that we get from this structure is that HDCR is an enzyme-decorated nanowire. So we have this chain of iron sulfur clusters from the ferredoxins in the center, and this nanowire is decorated with the enzymes, the hydrogenases and formate dehydrogenases that do the hydrogen splitting and CO2 reduction. Okay. And using this information from the structure, Helge, who was a student in the group of Fokker-Müller, he did several experiments with mutants informed by the structure to disrupt the filamentation at different points. So, for example, he could disrupt the filament within a subunit or between subunits, and also he could disrupt the iron-sulfur coordination. And in every single case, we saw the enzyme activity was drastically reduced. These mutants, they would grow much slower than the wild type bacteria. And so filament seems to be very important for the activity of this enzyme, which is the most efficient molecule that can do this reaction. However, it wasn't clear how is this filament organized in the cell, or if they are even like forming filaments like we saw in the grids, whether this exists in the native cells. So for this, we did focused ion beam milling of TKV cells, and this was our makeshift anaerobic environment for freezing these grids. It's not as fancy as what Bonnie showed you earlier today. And the idea here was basically that we had evaporating liquid nitrogen to drive the oxygen away. And we were controlling the environment using a candle, hoping that it would not light up. So this was almost 2 years ago. Nowadays, our collaborators, they have a much better environment for doing these experiments. And what we got from these feed mute cells, we acquired tube series. This was the work of my colleague Wojciech Witriński. And this is how our tomograms look like. What I'm showing here has been denoised using cryocare. And if you look towards the poles of the cells, it's not always at the poles, but most often, we have these strings of beads. And then we suspected that these might be the HDCR filaments. And importantly, we have also imaged the mutants where we have deleted the HDCR gene, and we could not find these filaments in the mutants. And so I set out to do subtomogram averaging of these filaments. And this is how the pipeline for processing this data looks like. I will not go into details here, but this was my very first subtomogram averaging project in Ben's lab. And my colleague Jonathan Wagner from the Max Planck Institute in Martinsried was teaching me how to do this. And most importantly, the key tools here were the packages TomoMan and StopCap, developed by Will Wan and Sagar. And TomoMan is basically a collection of MATLAB scripts to do the tomogram pre-processing, so the drift correction, the CTF estimation, tilt series alignment, and reconstruction in a very automated way. And StopCap is another package for doing subtomogram averaging. It's also based on MATLAB, and it's quite powerful. I know this might be a very unconventional pipeline compared to other things that I have seen here, but we like these tools because in our experience, StopCap can crack very difficult subtomogram averaging projects. And these things are open source and they are available on GitHub, but they are not very well documented yet, but we are working on it. And Sagar and Will are planning to release these tools sometime later this year with a publication and documentation. And I have also done some customizations to the StopCap code that we are now merging back into the main code and writing some tutorials. So stay tuned for updates on this front later this year. And how I started the processing here was I did a manual filament tracing in IMOD. This is similar to what Sam showed today for the IFT complexes. So I traced a few filaments by hand, and then I oversampled these filaments using spline interpolation in StopCap. And these filaments are quite difficult to visualize, even in the denoised tomograms, because they are very thin at this magnification, and they are very intertwined. So it's hard to see exactly where a filament starts and where it ends. But from this initial manual picking, I could get this average. And then I use this average to do template matching to hopefully recover or detect HDCR in the rest of the data set. And this is how the score maps from template matching look like. And here it was interesting to see that there were clusters of high scores. And so I used a little bit of scripting with Sagar, the Sagar plugin in Camera, to identify these clusters of filaments and mask only these parts of the score map. So I would basically filter out all the false positive peaks using this procedure. And this was this is almost entirely automated. I know. So if you have a difficult template matching case that for filaments, this is something that might help. And I would also be interested in trying the GRACE graph picking based tool that Bia presented yesterday. So what we got from this subtomogram averaging are these filaments resolved to about 17 angstroms. And we could see that the nanowire core fits relatively well in this map. However, the decorating enzymes are not as well resolved. And there are different possible explanations for this. Could be that one thing is that these filaments are flexible, so they bend in different ways. So this degrades the average, of course. And another thing might be that the stoichiometry is not complete. So might be that not all enzymes are present all the time along the filament. But what was most interesting here was to see that the nanowires, they are not alone. They are surrounded by neighboring filaments. So I always got in my averages this bundle of filaments. And this was very intriguing to us because we did not know that there was any sort of a lateral bundling in HDCR. And we could see after fitting the model in this average shown here that the helical parameters are quite different in vitro and in situ. And this might be because in the cell, these lateral interactions are enforcing it into a different conformation. But overall, the architecture is the same. And what was very nice as well, when we mapped back the particles into the tomograms, this was quite surprising to see that HDCR forms these large structures attached to the cell membrane, which I'm showing you here in this movie. HDCR is shown in yellow, and in blue we have the ribosomes, which I have also averaged from this bacterium, and we have the S layer in purple and the plasma membrane in gray. And this was completely surprising to us to see this higher order organization of HDCR filaments inside the cells. And we are not sure yet why does it form these large circular bundles that we call the portals, but we think it might have to do with some concentration mechanism to increase the efficiency of the reaction even further. And sometimes we would see full, complete round portals attached to the cell membrane. Sometimes we would see only partially assembled portals. Might be also that the feed milling procedure chops part of the portals as well. But this was something that we could only observe using in situ electron tomography, and quite surprising. And this was done until about 1.5 years ago, more or less. And after that, more recently, I think Reliant 4 was not available at the time that I started working on this project, but later I could learn how to use Reliant 4 and I could reprocess this data. And now I could push the resolution of the HDCR filaments to about 13 angstroms. We are now working on fitting the models into this map. This is still work that is ongoing. And it's very nice to see how just with new methods, we can squeeze more information out of the same data set. This is from about 11,000 particles from 17 tomograms. And another thing that I have processed from this same data set are the ribosomes from TKV, and also using Reliant 4. And the ribosomes here, they reached the 7 angstrom resolution, which is the limit for this data set. So this validated our pipeline very well. And the model that I'm going to show here fitted into this map is actually another bacterial ribosome that I did a flexible fitting using NemDnator. So please don't take this model too seriously here, because it's not the ribosome from this organism, but it's very similar. Finally, another interesting thing that we found in this data set, well, there are many interesting things to see here. We also have chemosensory arrays, the S layer that I've mentioned before, some interesting membrane division events, but we are also very intrigued by these large filaments that we see here more or less in the middle of the cell and close to the HDCR filament bundles that we don't know what they are. It's not HDCR, it's much bigger. And then I also calculated an average from these filaments. We call them the train tracks because in the tomogram slices, they look like rails. And this is how the average looks like. It's now at about 16 angstrom resolution. Sorry. Yeah, it's now at about 16 angstrom resolution, and we have no idea what this is. If you have seen something like that before, please tell me. We would be very curious to know. But we have now a new postdoc in the lab, Katie McCafferty, who is an expert in integrative modeling, and she will be helping us push visual proteomics of all the things in the cell. And she is trying to identify what this complex is and to build a model into it. So to summarize the HDCR part of my talk, I have shown you that the cryo-EM structure of HDCR explains how the filament is connected, and that this connectivity in the filament is key to the efficiency of the enzyme. And using a cryo-electron tomography, we could see that these filaments, they form bundles and portals, the large circular structures attached to the cell membrane. There are now several open questions that we would like to understand. For example, whether there are lateral interactions between the filaments. Also, how is HDCR anchored to the plasma membrane? There's probably some other complex in there that we would like to resolve. We would like to understand whether these HDCR portals are organizing metabolic subcompartments within the cell, and whether these other large filaments, the train tracks, whether it is interacting with HDCR, whether it's part of the metabolic pathway, or what is it doing there. And finally, we would like to know if we could use HDCR to do carbon fixation or to store hydrogen as a clean energy source. Now heading to the second part of my talk, still in the same data set from TKV, I mentioned that we see several interesting membrane events in there. So there are some cell division events, and some compartment formations. And if you are interested in these things, I recommend you to take a look at this data set. And we have a new membrane segmentation tool in the lab that can very accurately segment plasma membranes. And here are just some interesting examples from this data set where I have used this tool to do automatic segmentation. This is work from our student Lawrence Lam, who is shared between our group and the group of Qingying Peng at Helmholtz AI in Munich. And this is a very fruitful collaboration where we have developed and are developing new deep learning tools for analyzing cryo-ET data. For example, together with Emmanuel Mobile, Charles Kevran, and Antonio Martinez Sanchez, we have participated in the development of DeepFinder, which is a tool for picking particles in tomograms. And Lawrence has also developed a membrane, which is a tool specialized in membrane particle picking for tomograms. And membrane expects you, the original version of membrane expects you to have the membrane segmentations already. And now we are working on a new version of membrane that will also do the membrane segmentation for you. And this is still ongoing work, but we plan to release it later this year. You can already have a look at the code. It's on GitHub. And the plan is to have a universal and fast membrane segmentation tool, to have an easy to install package. It's a Python package. It's a neural network, which is a UNet as usual. In particular, this was inspired by NNUNet, which is a very powerful neural network for segmentation. And the idea of NNUNet is that it's a self-configurable architecture that will figure out the best network architecture for your particular problem. In our case, we are taking the best, the most interesting parts of NNUNet for our problem and building a slightly different neural network that is specializing in detecting membranes in electron tomograms. And the idea is that if performance is not satisfying using the pre-trained models, that the users should be able to easily do corrections and retrain the model. And we are also planning on having a platform where users can share their retrained models, and we can like pull models from the community to achieve even better performance. And we are also working on an Apari plugin for membrane seg with help from Alistair, Kevin, and Ben Barad from the group of Daniel Grotjen at Scripps. And yes, everyone is welcome to take a look and give it a try already. We are happy to help to help you using this tool. And whether you want to share some data with us to train the model on your data, or if you want to try it by yourself, we would be happy to hear your experience with it. And one thing that we are being very careful about is annotating the ground truth. Of course, nowadays we have these extremely powerful deep learning tools that are very impressive, but we often overlook the human side of training these models. And the performance will only be as good as the data that you train the network with. And we are lucky to have a great student, Simon, who is now working on annotating ground truth for training membrane seg. And here in this example, you can see that, so the network works on patches of the tomograms. So after running a prediction, Simon takes a look at the prediction and he manually annotates corrections. So in this example here, we have in red is what was already predicted. In blue is what should be added to the segmentation in the next training. And in purple is what, because of the missing wedge and noise and some other imperfections in the data, sometimes it's hard to be sure whether it is a membrane or not. So these parts can be ignored. So they will not influence the network in any way. And we also have another class, which is not explicitly shown here, which is background, which is what should definitely not be segmented. And here's one example of how just correcting a few patches drastically improves the performance of the network. Here's an example on mitochondria. And just by correcting, I think it was only 7 patches in this case, drastically improved the performance to be almost perfect. And why are we creating these tools? Is going back to the start of my talk, we are interested in understanding carbon fixation and other processes in the cell. And nowadays with automation, we will have much larger data sets to analyze that we want to study in a more or less automated fashion, because also like Tristan mentioned in his talk, with the scale of these data sets, it's becoming almost humanly impossible to manually look at the data. So we need tools to help us. And here are 2 examples of mitochondrial segmentation and thylakoid membrane segmentation. Oh, sorry. Yeah. And all these segmentations were done with membrane seg, which will be part of membrane version 2. And yes, like I said, if you want to try it or contribute data for training, please get in touch with us. And finally, I would like to thank all my colleagues in Ben's lab, especially Wojciech for all his work in the HDCR project. Also our collaborators, Jan and Anuj in Marburg, and Volker and Helge in Frankfurt. I would also like to thank Ting Peng and her group in Munich for helping us developing these new tools. I would also like to thank Will for all the help with Stopgap. Part of this work was done while we were still at the Max Planck Institute in Martinsried. So I would like to thank my colleagues from there. And yes, also I'd like to thank our high performance computing experts in Basel, and also all the help of #TeamTomo. It's a great community that yeah, it's a pleasure to be part with. And I would also like to thank the artists who have made beautiful renderings of the HDCR portals. Thank you very much.